So this is a follow-up session of a previous event titled Agrobacterium-mediated transformation of rice from mature seeds, a stalwart protocol of cereal transformation. Because this is a follow-up session, we won't be going through the protocol. Instead, this is a time for questions for Albert and discussion with the audience. I would like to reintroduce Dr. Albert Kausch. Dr. Kausch is a professor at the University of Rhode Island in the Department of Cell and Molecular Biology with a research focus on molecular improvement and gene discovery in plants. Albert was an early pioneer in plant transformation with now 40 years of experience in plant transformation technology development for commercial and academic research, as well as agricultural biotechnology. His broad industry experience extends from working with large multinational seed companies in a vast variety of areas. He maintains several active research collaborations on plant transformation and genome editing technology development through his laboratory at the University of Rhode Island. Dr. Kausch's contributions include technology and events generated through genetic modification, traditional breeding, genomics, and gene editing of various crop plants that are now grown commercially worldwide. So with that, let's get started. Questions will be taken from the Q&A box, and we will also take questions from the audience verbally. So to ask a question directly to Albert, please hit the raise hand button in the Zoom controls. I'll unmute you, and then you can ask your question. So please ask your questions as you think of them, and feel free to comment on other people's questions in the Q&A box. Any information presented today will be available on the Plantgene website along with a recording. And with that, I'll get us started with some pre-selected questions from the registration, and I'll turn things over to you, Albert.
Great, great. Hey, first, thanks for that introduction, and to everyone else, welcome back if you saw the first one. And I'm really happy to be here and really interested to hear your questions.
Thanks. So let's start with oh yeah, sorry. I will put a link in the chat for anyone who wasn't able to um see the first recording. It's available online. Definitely recommend that you go and watch it and read through the protocol as well. So I'll start with the first question. So how do you manage agrobacterium to ensure that there is no excessive growth? It can be quite stubborn to deal with.
Yeah, this is a good question, not only for rice, but just generally agrobacterium-mediated transformation. Um, Overgrowth can actually result from several factors. It depends really a lot on what strain of agro you're using. So commonly we're using EHA strains, LBA 4404 and AGL1. These all have different growth characteristics. If you're having problems with overgrowth, I would suggest looking at the OD, making sure that you're starting with around, we like to start with around an OD of over 400, maybe 500, maybe 550. But then you can also adjust your time of exposure. Many protocols will recommend 3 days of co-cultivation. If you're having overgrowth in that time, you can definitely cut that back. So you can control overgrowth by limiting the concentration of agro, limiting the exposure and co-cultivation. And I think using Phi- strains have also been really good at controlling agro overgrowth afterwards. Not available in all strains, but that's a useful tool. And then again, also the antibiotics that you use to control them. Cefotaxime and carbenicillin are a good combination, but different labs, you can read up on that and see what's best for your system. They're good questions. Thanks. I hope that helps.
Yeah, does anyone have any follow-up questions on that? If so, you can raise your hand, or you can just write in the Q&A box. So just while, yeah, again, if there, I'm going to move on to the next question, but feel free to write in your questions at any time. And so the next question was, how can we regenerate the transgenic plants in a shorter time? How do the culture conditions, example, temperature, humidity, light, impact callus induction and regeneration?
Yeah. I was telling Amy before we started, this is a great question. And again, not only for this protocol on rice, but for other monocots as well. In fact, in my lab, we've been working on truncating the sorghum transformation pipeline. So this is always on our mind as we want to increase throughput. Um, in sorghum, for example, just over the last couple of years, we've compressed the timeline from 12 months down to 24 weeks. But with that said, let me say this. You can't make a plant grow faster than a plant can grow. And that's about where you're going to be stuck. But there are some points that you can put pressure to to accelerate the timeline. Um, Gosh, mainly that is in the selection and regeneration phases. Uh, it would take me a while to explain this, um, but and I didn't talk about morphogenic growth regulators in the first workshop because it really pertains to a standard method. But the use of baby boom and mushel and GRF GIF can impact these systems significantly. Um, so they can come to bear on compression of the timeline too. Um, I hope that helps. Uh, but yeah, you can look at, I think, the each one of the areas that are able to be compressed, like how short of a time can you get away with selection, and still recover clones that do not have escapes. So it's things like that that come to bear on this. Um, Adjusting things like humidity, um, yes, you can optimize those, but those have pretty much been optimized by people like me already. Um, you know, I think compression of selection and regeneration times and using morphogenic regulators might help you get there. I hope that's helpful.
No, that's a great answer. Yeah, like we were talking about this question a little bit before. And yeah, I think that's a great point of there's all these things you can do that kind of build up and make a big difference. But that's a good point about you still have the plant has to take time to grow.
Yeah,
So there,
yeah,
yeah, there
yeah.
is a limit here.
Hey, to add to this though, we recently, with sorghum anyway, when you reach a plantlet size in tissue culture, your time to flower should be roughly the same as a wild type plant, as if that thing coming out of tissue culture, and that's another place you can accelerate and optimize. Anyway, right.
So we got some questions coming in. So people asked questions in the chat, which is fine. We're going to get to those. But if you could ask questions in the Q&A box, it's easier for me to keep track of what's been answered and what hasn't been. So I'm going to start with this question from Benjamin Kari Kari. Albert, have you ever worked with Ruby Reporter? If yes, on which trait, if root system architecture, does Ruby system affect on root development?
Oh, you know what? Bummer. I've not worked on Ruby. I'm
Okay.
working on M Scarlet right now, and it's hot. It really looks great in roots. So, but no, that is really, you stumped me on that one. No, I have not worked with Ruby.
All
Wait a
right.
minute. Wait
Yeah,
a
if
minute.
anyone
Wait a
else
minute. Wait a minute. No, wait. Yeah. No, DS Red. Sorry. No,
Oh, okay, yeah,
no
different
Ruby.
different system.
No.
Well, if anyone has worked on Ruby, you can uh, comment on people's questions. So feel free to comment, and you can share your experience with Benjamin if you'd like. Again, you just go to the Q&A box. There's a tab that will say answered, and you should be able to write. If you are having issues, you can let me know in the chat. So, this is a question, to what extent does the presence of aggro affect the germination of seeds that have been transformed?
It's the, well, let me,
So I think this is about,
sorry.
they did make a note that they did not attend the last session. So, which is totally fine. If you weren't able to, there's a recording on our planting website. But maybe you can just explain we're working with callus or yeah.
Yeah, so the plantlets recovered after this, and the seeds that come out of T0 plants function normally if the plants have been healthy all along. So there's no aggro present at that time. In fact, um, you're probably you can't PCR for your gene of interest in tissue culture because of the persistent presence of agrobacterium, if that's what you're referring to. But that's that.
Okay. So this is a question from Hope. Why are you using carbonicillin and cetozamine to, sorry, I probably mispronounced that, to control the bacteria instead of just one of them? At what point should I stop adding antibiotics to my media? In other
Oh,
words, should carbonicillin be added to regeneration and rooting media?
yeah, great question. Um, Actually, the acetosyringone is the signal that stimulates agrobacterium. So, antibiotics, um, you can use the common ones are cefotaxime and carbenicillin in some cocktail. Timentin is also useful and in some studies seems to promote shoot regeneration. Um, uh, I would stick with the recommended concentrations of kill. I know these things are expensive, but agro is as persistent as Lyme disease. I don't know if you know anything about that, but anyway, it's hard to kill. So you would want to retain the antibiotics uh, through, I'm sorry to say, regeneration, because it will remain persistent in pockets of tissue or even in uh, developing xylem and can contaminate your cultures later. So I would rather be safe than sorry. People soak them in agro. I mean, soak them in, they'll take callus. And actually, I don't think that's necessary. We haven't had this type of a overgrowth problem. Somebody out there should invent a suicide gene for agro. Thymine would be okay, but how about a switch? We just turn the thing off and kill it. This is a 20-year-old idea. I always thought somebody else was going to do it, so I never did it. So somebody, it's
No, that'd be huge.
right, right?
So yes, yeah, anyone on the call wants to get on that.
Save us all from
We'd all be
cefotaxime.
grateful.
Yeah, man, kill it. I mean,
All right, so this is a question from Joanne. Is there a good way to predict if a cassette will successfully transform a full left to right border insertion without rearrangements?
whoa. Um, Great question. Um, Not without trying it, I'm afraid. And this is a problem. Um, you know, in fact, vector to vector analysis is difficult. If you look at people try to test these things, pre-test them in protoplast systems, it's a struggle we have here too, still. vectors that don't work. How long do you have to go through this process to find out that it's not working? And but to the comment about rearrangements, there are some things that you can do about that. Um, Vector size, um, limit that to the cargo that it can carry, but also reiteration of promoters and termination signals should be avoided. You know, I I I I think there's some correlation with this. So I think things like that can help, but to pre-test that, other than a protoplast system, unfortunately, it's just a lot of work to do that, and we struggle with this. But this goes back to my pipeline analysis, right? You might want to think about this. We're constructing this in sorghum right now so that in the first 12 weeks, you can get to a plant that you can isolate gDNA and screen that for your single gene copy inserts, integrity, and everything at about 12 weeks in sorghum. And then at that point, fail fast, and get rid of things out of the greenhouse. So there you go. Something like that.
Thanks. Yeah, you brought up a really good point because this is a conversation I just had recently about whether, yeah, doing protoplast assay or to test your vectors or to not do a protoplast assay. Yeah, I think some people recommend it, but like you said, it can be very time consuming and stuff. So you guys don't do a protoplast
We
assay
don't right now.
for okay.
Testing guides. My collaborators are testing guides on protoplasts, and that's good. That's
Okay.
but still, you gotta do it.
Hmm.
You gotta just see how it works.
Okay, we have a question from Hope. Hope, if you can just unmute and you can ask your question directly.
I hope.
Hi. Thank you very much for this follow-up. Although for the follow-up session, although I didn't join the first section, I was the one who asked the question about using carbon cefoxazine and carbonicillin. So there is a part of the question that you didn't answer. So the question is, should we use both cefoxazine and carbonicillin in media, or to wash off the agrobacterium from cali, or we should just use one of them? If we're using two, what's the reason behind using both antibiotics? Thank you.
Oh, that is a really good question. Why use both? And again, this is because they're expensive. We have tested them both independently and found that the combination um, seemed to control things better. So maybe it was over an abundance of care, so that you may be able to get away with one or the other. When we were using Timentin was sufficient on its own. Um, I think we did that because the combination was more successful. I think there are literally hundreds of rice protocols at this point you can look at, and many of them are some sort of iteration on this mature seed um, combination. And one that I saw takes the callus and puts it into a suspension and does the infection in a suspension. Remember, in my protocol, we're spotting it. So I think in the method of spotting it, the way that with a micropipetter right onto the callus, you can control how much agrobacterium is actually getting on the callus. Um, and so I think the immersion method seems to me like it's a lot more work, but also really messy for getting rid of the aggro. Um, I'd have to say you'd have to try that side by each. Sorry, I don't have a better answer for that one.
Mm,
Thanks, Hope.
mm.
So this is a question actually, again, we were talking about before we started today's webinar. This is from Abida. Hello, I was wondering if you have the basic stock solution formulation of CHU N6 medium that you used in your protocol. Unfortunately, we currently don't have it available here in my lab.
Wow, that is a great question. And we were talking about this prior to this. Um, oh, I could put this on a shared document, but do you want me to do that? Do you want me to show you what this is?
Yeah, sure. If you want to share your slides.
We could do that. Um, this is an interesting question as it relates to this protocol. Um, why use N6 in the first place? Can everyone see this okay?
Yes, yeah, we can see it. The PowerPoint slides, it's not in presentation mode.
Yeah. Oh, you can see it in a PowerPoint slide though.
Yes, we can see it.
Okay, that's
Yep.
good. That's good. Then I can flip around things. If you can see that, okay, this is the N6 media, and in comparison to MS, and here are the ingredients. You should be able to get this, I think, from Phytotech, but you could look this up and make it from scratch. But what I'd like to point out here is the difference is what is responsible for encouraging early embryogenic callus in rice using N6 because notice it has no ammonium nitrate and it has 463 milligrams per liter of ammonium sulfate. And N6 is higher in potassium nitrate. These nitrogen levels control morphogenesis in plants generally. And in rice, this is really quite spectacular in arresting this callus at a very early stage of embryogenesis. And this is why this protocol is so successful. You know, when you transform one of these cells, and then you're selecting by killing all of the remaining untransformed tissue around it, that single cell has to be able to reiterate this culture, remain totipotent, and regenerate. That's asking quite a bit. And the reason that you can do this is those single cells are not reliant on each other for their survival, as would be the case in a more organized tissue system. So this is why N6 is truly very important to this protocol. It also has no myoinositol, which also favors late stage differentiation more towards regeneration phase. And N6 is higher in thiamine. These subtle differences make a big difference in the early response of callus formation and why this protocol is as successful as it is for Japonica rice varieties. Anyway, that's my I hope that helps. That's my thoughts on that.
Thanks. Yeah, that'll be great. I'll have to get that slide from you and we can
Sure.
but this is from the master class, right? That you did for
Right.
Christian with Christian. Okay, great. So I'll
Oh, I
include,
think that slide,
yeah, was
I'm not
that,
sure that was in there or not. I might have to send that to you, but
oh, okay.
you know, it's a good one if you're teaching tissue culture or plant physiology. You know, these are not hormones, these are micro and macronutrients that are directly involved with morphogenesis.
Great. Hope,
Thank you
do you have
again.
another question? Yeah, go
All
ahead.
right.
Sorry.
Yeah. So, I want to ask the host, have you I know you work with rice, so you must have gotten some regenerated colors, correct?
Yeah.
Okay. I've been having this challenge of having regenerated color in my regeneration media. I followed for some time. I would follow some protocols and it won't give me the expected result. And recently, I just changed my protocol to the newer one that has about 80%. I think they mentioned 80% transformation efficiency
Yeah, great.
in the paper. So I'm following that protocol, but the protocol still does not include carbonicillin in the regeneration media. And from what I read, I saw that carbonicillin reduces regeneration rate in the regeneration media.
Uh-huh.
So I'm concerned as to how do you get regenerated calli? Because I've done everything possible and I still haven't gotten regenerated rice calli.
What variety are you working with?
I'm working with Nico Bear.
Oh, okay. And you're having problems with the regeneration prop.
Yes.
Hmm. Um, I'm thinking it's not the carbonicillin that's inhibiting you. Um, um, I would have to know. So you're getting so let me ask you, what is your selection? Are you using hygromycin?
Basta selection.
What's this?
Basta selection.
Okay. And are your colonies clean at this point?
Yes, they are. So I
So,
culture a single colony and that's what I
yep.
use for transformation.
Yeah. Okay. And then you're moving it onto you're dropping out the auxin and you're moving it onto regeneration and into the light, right?
Correct.
And you don't see any greening.
No, I don't. So it only gets whitish and whitish. That's the color. So it's like it's generating a secondary color, but the secondary color never gets to form
Never.
any greenish shoot. Yeah,
Uh-huh. And gosh, it's hard to diagnose this without seeing it. Um, I am with Nippon Bari, this generally okay, so how long have your colonies been in selection?
Um, so I do select for 2 weeks.
Yeah, this should be okay. Prolonged selection and prolonged time in tissue culture will eventually result in reduced regeneration. And this is because non-regenerable tissue grows faster in pretty much all monocot systems. Unless you visually subculture, maybe what you could do is that with your colonies that you're putting onto regeneration, um, we used to move the entire colony. You could go in with a dissecting scope and select out the most mature looking tissue and move that onto your regeneration media. That might help you. Um,
Okay,
I'm just now um, so the regeneration media is switching over to MS media, correct? You're doing,
correct.
yeah. And I'm just looking at this. And you're using carbenicillin and timetin or just carbenicillin?
Just carbonicillin. That's for the new protocol.
Yeah,
Sorry,
timetin.
that's for the old protocol. Yeah.
Oh, yeah,
The new protocol says I shouldn't use carbonicillin at all.
yeah. Timetin might, but timetin hasn't helped you out, huh?
No, we never got to use simmering. We've only been using carbonicillin.
Yeah. You know what? In our practice, that really didn't inhibit regeneration, though. So you're dropping out the 2,4-D, um,
Hmm.
and you're adding NAA. Is that correct?
Yes, correct. Hmm.
Yeah. Um, Gosh, I don't know. I don't know what could be going wrong here, I have to say.
Okay, so this is what I have in my regeneration media. I have an MS media with vitamins. I
Yeah.
have sucrose, and then I'm using the regular plant agar. And then I have my selection. So for Basta, I use half strength. And then I use the BAP, then I use carnitine, and then NAA. So that's what the new protocol gives me.
Yeah.
Uh,
So your
I,
kinetin is about 2 mg per mil.
no, 1 milligram per liter.
Okay. That's a yeah, I meant 2. 1 milligram per liter. Yeah.
Yeah,
You could increase that to 2. But I'm thinking actually, so you have 30 grams of sucrose.
correct.
Have you included sorbitol?
No, sorbitol, no.
Oh, that would help you tremendously.
Oh, okay.
30 grams of sorbitol will help mature your embryos.
Okay.
And
30 grams per liter.
yes, I think we've hit on it. Hey, this is in my protocol. If you send me an email, I'll send you the PowerPoint that goes with this. Or it's Amy, I think this is posted, right?
Yes, yeah, the protocol is posted.
Yeah.
Let me put a link
I
in the
think we put
chat.
our finger on it. I think it's these
I think it might be the
sorbitol
sorbitol.
to help you out a lot. And let me ask you
Okay.
also, are you using gel right, or what's your gelling agent?
It's a plant agar.
Yeah, I
Um,
would go with gel right
okay.
if you can get gel
Gel
right.
right.
Yeah,
Okay.
yeah. I would say refer back to this protocol and
Okay.
look at any deviations you've made from this and see what you can adjust.
All right.
I would
All
stand
right.
by this one. I've played around with this, and usually things I play around with don't work out so well. So
Okay.
you know, so you can think about sorbitol then as functioning in a way to mature the embryo the way a zygotic embryo would mature in a seed. It's
Okay.
sort of desiccating it in a way, having an osmotic component that's driving differentiation.
Okay.
All of these things are geared towards developing normal plant development and how you steer that.
Mhm. Alright. So your protocol, how do I get it?
So I just put a link in the chat.
Okay.
So you can access the protocol and then you can also access the recording from the previous event.
All right. Thank you very much.
Yep. Yeah, you're welcome. Good
Hope
luck with
that
your
helps. Let me
transformation.
know how that works out for you, though. Okay, you have no
Sure,
problem.
sure,
Yeah, yeah.
sure, I will. Thank you.
Yeah, Albert, do you mind if I put your email in the
Oh, by
chat?
all
Is that
means, go
okay?
ahead. Yeah,
Okay. All right,
yeah.
great. I'll do
I
that
love to hear
too.
from people.
So you can email Albert here. So I just put it in the chat. So it's apcouch@uri.edu is his email.
Mm.
All right. Yeah. Thanks again, Hope. Good luck with your transformations.
It'll
Thank you.
work.
Yeah, you'll get there.
It will work.
Thank you.
Yeah.
So this is a question from Abida. How can I understand if I'm infecting embryonic callus if I don't have a dissecting scope?
I love this one too. You saw that image I just projected about early embryogenic callus. In this protocol, this is quite remarkable. When you dehusk these seeds and you put them onto the callus induction media, in the first 5 days, I'm going to say this, pretty much the only thing that comes out of this is early embryogenic callus. This is not like any other monocot I have ever seen under any other circumstance. And I am a strong proponent of visual selection, especially in other monocots like maize, where you can visually see the early embryogenic callus under a dissecting scope and tweeze it apart and train your callus to remain in that mode. You can do this for other monocots. In this situation, you don't need to do that. If you take this as it emerges from the seed and you're going to, clip off the epicotyl and the radical and let that callus mature only 5 days to a week, or you can subculture this for up to 3 months. And I do not think we used visual selection to do this. If you regularly subculture this stuff, it is going to maintain as an early embryogenic callus for quite some time. I wouldn't use it much longer than 3 months. You'll start to get somoclonal variation, but you can start your callus fresh. Everything that comes out of that should be. I like the spotting method for this. You could just spot right onto the isolated callus, or you can bulk up the callus and then use it in an array and spot them. We were in terms of transformation efficiency here, we were determining this on number of transformants per callus piece treated, and that's a different way of maybe assessing it. But so frequencies over 80% here are not uncommon, even with other genotypes. I think the one I posted was on Nippon-bari, but this also works on Taipei 309 and Taki for other japonicas. Anyway, that's my thoughts on that.
Kind of, I guess not really a follow-up, but people had some questions about chimeric
Yeah.
Or chimeras. So one person had asked, how do you know if you're working with chimeric tissue? And then another person had asked, sorry, I'm trying to scroll to, so this is from Ruby. Can we add a resting phase after co-cultivation to get more transformants and then put it on selection, or would it give a lot more chimeric tissue?
Okay, yeah, this is about chimeric tissue. So this really relates to selectable markers. If I have recommended here either bialofos at 3 mg per liter or hygromycin. So hygromycin is an extraordinarily clean selection system for this. It's going to literally kill everything around that that's not transformed very quickly. And because of this system, the early embryogenic callus will grow back as it should be a homogeneous callus line. Nothing should survive hygromycin selection. You should get 0 escapes out of this protocol. With bialofos, it's a slower kill. Remember, bialofos kills by inhibiting glutamine synthetase, which causes ammonia to build up in the cell. So you're actually killing your non-transformed cells by accumulating ammonia. Now, in that milieu, your transformed cell is seeing a ton of ammonia, right? So again, if you subculture that resistant colony to homogeneity, you should not get any chimeric tissue whatsoever. If you're getting chimeric tissue, it's either not long enough in selection, you've relieved selection. Don't take it off a selection like ever, even in regeneration phase. You're trying to kill non-transformed plants, not save them. I mean, as the tissue culturist in us, we want to save everything. No, you don't. After co-cultivation, I think, callus induction, co-cultivation, and then a resting phase is important. And I really dislike the term resting phase as if they're sleeping. Come on, they're actively growing. You're trying, think about the biology here. You're going from a single cell, and all of these are generated by a single cell, by the way. We know that from southern blots. Every event is originated from a single cell because all clones have the same southern blot. So you got to time this in such that one cell grows into a cluster, grows big enough to undergo selection and self-sustainability. So those are the constraints that you're working with. And resting allows that colony to get big enough to survive the death of the surrounding non-transformed tissue when it goes into selection.
Thanks for that. And yeah, that is, other people had this question too, because I think we did talk about that a bit in the original, was chimeras and how to avoid it. And yeah, I think you made a really great point about the resting. It kind of makes it seem maybe not as important of a step, or that, yeah, but it is. It's really
Take a break.
important. So don't skip that. Um, this is a question from Don Ping. Do you see any detrimental effects of developmental regulators on transformation?
Love this question. In fact, I think we were the first academic lab to use baby boom and mushel in maize and sorghum on recalcitrant varieties. Pertaining to rice, okay. I might have a slide or two on this. Not really too much that's helpful. You see that? Maybe.
No, yeah.
Wait a minute.
There we go.
Yeah, okay.
All right, it just started.
Morphogenic regulator-mediated transformation has been used in rice and extended varietal independence. And on mature tissue, not as useful. I think the recent paper out of Corteva on leaf transformation is spectacular in terms of its use on rice and indica varieties and all monocots. The problem has been translating this into public use. My lab, as well as other labs, are right now working on a publicly available variety of the leaf transformation vector. Now, this is really good because baby boom and mushel on leaves functions as a selectable marker. The only thing that possibly comes out of this are transformed cells. GRF GIF. So I don't know if any of you have experimented with this. In sorghum, this is fantastic. It is so let me just comment on this. Baby boom and mushel seem to be acting on the very early phases of transformation, stimulating the first cell divisions and very early embryo formation to the pro-embryo point. I've had discussions with Juan Debonardi out at out in California, and so the thinking is that GRF GIF is involved with later regeneration phases. So if you have early embryos that for one reason or another are unable to go through regeneration, GRF GIF seems to recover these and stimulate the number of plants out of it. Look into Juan Debonardi's 2020, I think it was a Nature paper, on this on wheat and how this has really been fantastic. And I can report the same thing in sorghum. Now, I have a collaborator who's done this and my protocol here on rice and has tried this on rice and does not see a significant beneficial effect to using GRF GIF there. I'm somewhat surprised by this, and I think that could be explored more. But I think morphogenic regulators, if you're going to get into this, and leaf transformation on rice. First of all, leaf transformation, great idea. I would suggest this, though, that this mature seed protocol is still easier for you to do than it is leaf transformation. Just practically speaking, you can grab a handful of seeds anytime and start this experiment. You don't have to prep the plants like you would for that. It depends on your needs, you know? So I'd say for recalcitrant varieties, exploring morphogenic regulators, especially baby boom and wuschel, but GRF GIF, great. I'd love to collaborate with you on it. Anyway, thanks for asking.
Thanks for the, oh yeah, so Dan Ping just responded in the chat. Thank you for your answer. Very helpful. That's good to know. Glad we answered your question. This is a question from Nikita. So this is about ryegrass. So I'm not
Bring it on.
sure. All right, great. Yeah, let's talk
The
about
monocot.
ryegrass then. So, hello, Albert. I am currently working on perennial ryegrass transformation. While I am achieving good transformation efficiency as evaluated using Ruby as a reporter gene,
Uh-huh.
I am facing challenges with transformed shoot regeneration from callus. The regenerated shoots consistently test negative for transformation, confirmed via PCR. I'm getting very good regeneration efficiency with non-transformed plants, so my regeneration media is optimized well. I've tried using various developmental regulators, including baby boom, wuschel, and GRF, GIF, but I have not observed successful regeneration of transformed shoots. Would you have any suggestions or insights that might help improve transformed shoot regeneration in ryegrass?
Yeah, I've
And
worked.
they mentioned, sorry, one more thing. So they mentioned they have tried this protocol with and without the developmental regulators.
Yeah, that's a good one. It's been a while. I've worked on perennial rye and other turf grasses, creeping bent, fescues, whatnot. And surprisingly, uh, in my experience with those, the variety dependence is not that significant in comparison to some other ones. So you might be kind of safe there. I'd like to know what variety you are working on, but I'll tell you, um, just from this explanation, um, I'm wondering if your selected callus, but if you're getting if you're getting escapes and non-transformed plants back, that would tell me that your selected callus is not homogeneous. So I don't know what your selectable marker is, but one way to overcome this problem would be to culture your putative transformants on the selection killing levels of the selection agent long enough, and it's got to retain its regenerability. So you might have to visually go in there and select for embryogenic callus. I don't know if you're familiar with that, but in practice, that would be the best way to do it. You know, there's another crazy thing I want to throw out to you guys since you're here. Visual selection. My entire career, visual selection on monocots has been attempted without chemical selection. Visual selection has really never been achieved. I mean, people have tried this all over the place, including myself. Can you just select out for a GFP colony and grow it? No, you can't. Try it. Um, there is one dubious paper on orchids where they claim to have done this, but I think these morphogenic regulators are changing this situation because you can go in and observe these very early stages in regeneration and culture those separately. So in your fescue experiments, I'm thinking you're losing regenerability of your, well, no, you say it's got to be your selection isn't clean enough, I think. That'd be one possibility. I hope that helps. But you shouldn't have any escapes at that point. Send me images.
All right. This is a question from Krishna. Despite establishing an efficient regeneration protocol and testing multiple factors that fail to induce infection, including virulence plasmids, different agrobacterium strains, and explant age, what alternative strategies can be employed to successfully infect plant tissues with agrobacterium for stable genetic transformation?
What's the system? Not sure.
Doesn't say, but Krishna, if you would like to add to your question or if you want to raise your hand, happy to get some more information. Okay, she just raised her hand. Hi, Krishna.
Hi, can you hear me?
Yes,
Yeah.
we can.
Very nice. That's wonderful. I'm really happy to be part of this section. So my question is, in general, is not just associated to the rice. It could be any monocot plant like oats, wheat, barley, or any plant species.
And so you're concerned with stimulating the agrobacterium to transfer tDNA. Is that correct?
Exactly.
Yeah.
Yeah.
So generally 100 using acetosyringone and priming both the agro and in your infection media. So having acetosyringone, and the acetosyringone must be made fresh from a frozen stock. So it degrades quickly. So you're making fresh acetosyringone in your infection media, and then some people you could include acetosyringone in your co-cultivation media as well. And then after that, I think if you're having a problem, then it would be with your agro strain itself.
I have tried transformation by using helper plasmids such as PKL 2299 and 22660A and
Yes.
also PSB1.
Yes. Did that improve?
No, the tissues are, I don't see any infection.
Okay, so how are you determining infection?
I have a GFP
Oh,
expressing vector as a control.
okay, so you should be seeing transient expression GFP hits,
Exactly,
but you're not seeing any GFP hits.
exactly.
Huh. Um, so uh, I would have to imagine. So you know that GFP is in your construct.
I always use tobacco as an internal control to check the transformation is happening.
Yeah.
And I always see a GFP expression in tobacco, but not in the tissue of my interest.
What is the tissue of your interest?
My tissue of interest is oats.
Oats. So, and then are you infecting oat callus?
I have tried several tissues such as leaf-based segments, callus with the different ages starting from 3 days to 40 days.
Uh-huh.
And then I have also used mature embryo, immature embryos as well.
What's the promoter driving GFP?
It's maize ubiquitin promoter.
So that should be working in oats.
Yes.
And it's working in tobacco. That's interesting. But so if you know that the construct is working, are you following a oat protocol right now?
I have recently reviewed a paper. There is no agrobacterium protocol is available for the oats. And there is one publication from the Polish, let's say, authors, but this is not reproducible. And this was the old paper.
Huh. I will have to look that up. And can you email me the paper that you have, and I'll take a look at that. And
For sure, I will send you.
yeah, let's try to work through this problem.
Yeah, exactly.
Hard to diagnose under a Zoom. Sorry.
Yeah, sure. And in general, what is your opinion on that? For example, you have a tissue and then you try to use all the available information, such as using ternary vector systems, supervirulence plasmids, and then new generation agrobacterium strains such as EHA with REC minus or LBA4404 thymidine and all the new
Yeah.
information. How can one find a solution to the tissues that are recalcitrant to agrobacterium or resistant to agrobacterium?
I think you could start, especially with oats, going to a protocol that is successful there. There are successful oat protocols. I haven't worked on oats myself, but I think to start there, it's what's the agro strain that you're using now?
I am using almost like I have used more than 5, but currently I'm using the one from the Paul Lawrence lab from the VIB Ghent.
Yeah, so that's an
It's EHA 105 REC minus, and then also the LBA 4404.
yeah. So those those should work. I think that the helper plasmids that are available for those strains also should help. I'm thinking, the reason I'm hesitating here is I'm thinking that in your case it's something really simple, and I'm trying to think of what that could be. But not exactly sure. So send me your protocol and maybe we can work this out.
Yeah, sure. And you mentioned that you worked with the oats.
No, I have not worked with oats.
Okay, got it.
Yeah.
Mhm.
It's one of the rare monocots I have not worked on, in fact, but okay. Yeah.
Cool, that's nice. Thank you. I look forward to send you this information.
Sure, we'll work it out.
Okay, so we are coming up to the end of the hour. I'm going to have this last question from Ian Curtis. Why can you not use kanamycin as a selectable marker in rice transformation? Is it true that rice has an NPT2 gene in its genome, which has been rumored? Interesting. I've never heard this before.
Yeah, the aminoglycosides in general, kanamycin and rice. I haven't heard the idea about having an NPT2 gene or analog there. Hi, Ian, by the way, and I got your email, and we can talk about that some other time. But thanks for the question. And yeah, I I had been a aminoglycoside skeptic in monocots for a long time, but now we can see many protocols that have been developed for the monocots. Just look at the leaf transformation paper. That's all based on NPT2 at Corteva, using G418 and analogs like that are more conducive to plant cell selection. So I would say never, Ian. Maybe that could work. I don't know. There might be some varieties, but I think maybe the selectable marker of choice there might be something other than an aminoglycoside anyway.
Right. Yeah, thanks for that explanation. Yeah, I wasn't familiar with that, so that's interesting. All right, so we are at the end of the hour. I did want to end with some comments that people wrote. So this from Ruby. Thank you, Albert. I heard resting phase from someone's presentation. I did not like it too. So yeah, maybe we'll change the wording on that. And then I wanted to just share this comment from Chandak Basu. Sorry if I'm mispronouncing that. I was Dr. Cash's student during my PhD.
Tell him hello.
Yeah,
Yeah,
hi.
great.
Um,
Hi, Shandak.
I'm indebted to him and learned a lot from him. His passions for plant biotechnology inspired me to become a professor of plant biology. And he signed it off, Chandak Basu, professor at California State University in Northridge.
Oh, thanks. Thank you very much, Shandak.
So yeah, that's great to have that follow-up. So, all right. Thank you, everyone. I think there were a few questions that we didn't get to. I tried to answer the questions as best as I could. If it was a question that we answered in the previous event, again, just encourage you to go check that out if you haven't. It's on our website, which is plantgene.sivb.org. And let me put that in the chat one last time. If you become a Planching member, you will get an email with the recording and with any of the other information presented today. So we shared a few slides like with the N6 media and such. That will all be posted on the website. And if you are a member, you'll get a reminder about it. So don't forget to become a member if you aren't already, and you can register at this link here. Just put that in the chat. So again, thank you everyone. So this is the second follow-up session that we've done in general, and we're always looking to improve upon it. So you can email us with anything that you liked, anything you didn't like, and those comments are always really helpful. We read everything that you send in. So all right. Again, just thank you to Albert for joining us for the second time for your follow-up session. We really appreciate you taking the time to share your expertise with us.
Thank you, everybody. Thank you, Amy.
All right. Well, thank you, everyone. Bye.
Bye.
PlantGene would like to thank the National Science Foundation for their support. To learn more, please visit their website at nsf.gov.